start just to say good morning. I hope you are all well, wherever you're joining us from. I'd very much like you to welcome you to today's talk. For those who don't know, my name is Richard Mackenzie. I'm based in London at Roehampton University, and I'll be hosting today's talk. My area of research very much fits in with the theme of today's talk, which focuses on insulin resistance and metabolism. Now, I've been looking forward to this talk for almost 2 years now. It seems like John and I have been discussing this, him coming to Roehampton for a long time. This is a great pleasure to have Professor John Hawley with us today. Like previous sessions, though, I'm just going to keep my introduction to the talk fairly brief so that we can spend more time listening to what John has to say. I'd be very much surprised if any of our guests haven't heard of our speaker today. His name is pretty much synonymous with the field of nutrition and exercise metabolism. And Professor Hawley is currently director of the Mary MacKillop Institute of Health Research and head of exercise and nutrition research program at Australian's Catholic University in Melbourne, Australia. He has published over 280 manuscripts, and that's growing pretty much every week, and sits on many editorial boards for international journals, including some that we are obviously very interested in, the American Journal of Physiology, Endocrine and Metabolism, and the Journal of Applied Physiology. His lab researches the interaction of exercise and diet on skeletal muscle metabolism and the molecular basis of exercise training adaptations, as well as the cellular basis underlying exercise-induced improvements in insulin action. Thus, his work very much fits in with the theme of the series of talks. So without further ado, it's a real pleasure to be able to welcome Professor John Hawley to today's session. John.
Thanks very much. Thanks, Richard. Thank you for the introduction, and welcome wherever you are. It's about 6 o'clock here in Australia. I've just finished an hour-and-a-half riding around the lake. I saw about 3 people and a dog. It's very strange at the moment. We're all in the same situation. You can see that I've gone for the George Clooney unshaven look. There's really no reason to shave because I'm not going out and I'm not seeing anyone. So hopefully we'll get something out of this evening's session. I saw the names pick up briefly on the screen. I saw a few that I should be very scared about in question time, including Professor Eric Richter, but hopefully we can go through some of the things today that are related to chrono-nutrition. So again, without wasting too much time, we were gradually emerging from the lockdown and Melbourne slowly returning to normal. Again, I'm not sure where you all are, which is where I am at the moment, 600 kilometers away from Melbourne. So I'm virtually spending my whole day talking to my students on a screen, and I must be honest, I'm completely over that, and we'll be looking forward to getting back to work and actually seeing people and, you know, having a proper discussion about science, which I hope we can do tonight. Some of you obviously review for journals and are in the same boat. I have never, ever in the past 3 months had as many requests to review papers. Most of them have the word COVID-19 on, and then most of them are reviews, and I guess 50% of them have been pretty ordinary. So it's been a time where people have obviously thought, well, now I can catch up with the writing. So I think this year and next year are going to be the year of the review or the year of the commentary. And again, if you've got COVID-19 in the title, you're probably doing okay. But not to miss out on that, and this is just to give you a little teaser, this was actually accepted this morning. This is a commentary in the journal Sports Medicine, which we wrote with Andy King here, a PhD student of mine, now a postdoc, Louise Burke and Shona Halson, and it deals with the challenges, if you like, of maintaining metabolic health during a lockdown period. I'm not going to discuss sleep tonight. I'm not going to really discuss physical activity other than in a couple of slides, but I'm going to discuss how this global pandemic...
John, just to say your slides aren't sharing, I don't believe. I know we did it beforehand, but I've just given you back host, so apologies for that. That's
all right. So now what do you want me to do?
Well, just go into Zoom again and hit share slides. So I was able to see them, but I don't believe anyone else was.
Okay.
So that's my bad, sorry.
Can you see them now? No?
Not yet.
Hello, just to say I can see them.
So can we see them now?
I can see them.
Okay, everyone can see them. Great. Thanks, John.
All right, take 2. We'll just start from where we finished. If you missed out there, you just missed a nice picture of Melbourne in the summer and a brief slide on the fact that everyone's publishing review articles now. And so we thought we'd be no exception. And as I just alluded to, I'm going to talk today not about the sleep and inactivity. These are covered in the review, which will be out in 2 or 3 weeks, but mainly about how the lockdown and certain things that we can do for nutrition may be able to help in these type of situations. All right, I'm going to start with a strange slide and a very, very old publication. This is even before your time, Richard, 1729. And this was really the beginning of circadian biology. And this guy, as were the scientists of the day, was an astronomer, a physicist, and a biologist. So goodness knows how you do all those three simultaneously. But he was really the first person to observe that there was a diurnal pattern where the leaves of plants opened during sunlight exposure and closed during the night. And he was the first to describe this in a paper which was 1 page and about 10 lines long. So obviously didn't incur too much in page charges there. And this is really the first example of circadian biology. And this was done in plants. Now, he was so confident of the fact that plants opened at various times of the day that if you look on the left-hand side of the slide there, and you'll see a myriad of different plants, and I am certainly no botanist, I have got no idea what any of them are, but they opened at discrete times of the day. And he actually made a 24-hour clock based alone on the opening and closing of plants and their leaves, which is fairly phenomenal, if not a little bit gross. And I doubt, you know, you'd be looking to calibrate too many things in the lab with a clock based on plants. And today, just for an example, if you look at NASA and the atomic clock loses 1 second per 10 million years. So probably a little bit more accurate, but 1729, bear in mind that this was really the beginning, if you like, of circadian biology. Now, bringing us, you know, very much up to steam very, very quickly, 2017 saw the Nobel Prize in Physiology awarded to these 3 guys for their work predominantly in fruit flies. And we'll get on to how some of the animal work may or may not translate into humans a little bit later. But this was a landshed, because if you look at PubMed after 2017, you will see that the number of articles with circadian rhythms, circadian biology, and the such like has just exponentially increased. So this is a very, very fertile area of research at the moment. And these guys, you know, several years ago were awarded the Nobel Prize for this. So this was, if you like, the watershed moment where people really started to take notice of how circadian biology could be interpreted, how circadian biology can be disrupted by certain nutrition or inactivity patterns or no exposure to daylight and such other factors that we'll discuss. And as I said, has led really to a lot of interest in this field by people who perhaps previously weren't interested in it. And I will include myself among those. I seem to have trouble changing slides here. Okay, just a little bit of a background. So in the hypothalamus, you've got this thing called the suprachiasmatic nucleus, or SCN. And if you think of this as mission control, you can't go wrong. This is the main controller of the circadian rhythms that you have on a daily basis. And there've been studies where they've taken people underground, put them in caves with no exposure to sunlight, and still circadian biology persists for about 6 to 8 weeks before things start going a little bit awry. And generally the circadian rhythms are around 24 to 26 hours. The main, what we call Zeitgeber, and my German's atrocious, but that is really just another word for Timegeber, is the exposure to sunlight and darkness. So that's really what the SCN is about controlling. It responds to sunlight and the cycles of light and darkness. Now that's mission control, but we'd also have what we call fine-tuning organs and tissues. So skeletal muscle, the liver, for example, the kidneys, and other organs in the body have their own internal clocks. And as you can imagine, these are responsive to other cues. And 2 of the most powerful are nutritional availability and activity/inactivity. So just to summarize this slide, because we need this understanding before we move on, the SCN really is mission control. That controls all circadian biology, and largely the cue for that is light and dark. However, these peripheral clues, and I'm going to talk more or less exclusively about nutrition tonight, are fine tuners. So the time of eating, the time of activity, these fine tune via peripheral tissues such as skeletal muscle and feed back to the SCN. So you can see perhaps how we can disrupt these. For example, now we're indoors most of the day. I know you're going into summer, but you know, summer in the Northern Hemisphere is a bit like winter in Australia and New Zealand. So we're still getting ample exposure to sunlight. I rode for an hour and a half today in the sun. Nothing wrong with my circadian biology. But I predict that, you know, in long winters, these things really mess up the SCN and really make a difference to circadian biology and circadian rhythms. So I'm going to ask you a question in a couple of minutes. I've already said that the timing of food is an important and powerful time giver. And the next question for you, and this is about all the homework that you have to do, is firstly, write down on a piece of paper how many of you have 3 square meals a day. And that is, you know, a traditional breakfast, lunch and dinner. And the reason I put that slide up is, where does this term come from? And apparently it comes from the British Royal Navy, and they serve their meals on square plates. So goodness knows how that translated into 3 meals a day or 3 square meals a day, but that's where the origin comes from. And the other question I'm going to ask you is, what is the duration of your daily feeding cycle? Now, during the lockdown, I'm acknowledging that this is not normal, but perhaps think back to what we were like 3 months ago. And I want you to think of the time of your first energy intake, the first nutrient intake of the day, which is obviously usually breakfast. Breakfast could be any time between 4 o'clock and 10 or 11 o'clock, and your last energy intake of the day. Now, I want you just to compare that and see if it's changed during the lockdown. And I'll give you some numbers in a minute, which are pretty startling. All right, so the first intake of nutrients throughout the day and the last intake. Water doesn't count, coffee alone doesn't count unless you've got milk or sugar in it. And write down the number pre-COVID-19 lockdown and post-COVID-19 lockdown. The other thing that I'm not going to talk about today, because in the interest of time, and it's really a whole nother lecture on its own, is how the timing of exercise throughout the day is also an incredibly important Zeitgeber and has profound implications on things like insulin sensitivity, particularly in skeletal muscle. We haven't got time to talk about that today, but a recent review article, which we published in Exercise and Sports Science Review, which came out just a couple of months ago, does discuss the appropriate times to eat and exercise. But I might add, just in passing, before we dismiss exercise completely in this lecture, that there aren't that many studies looking at the optimal time to exercise. And of course, one size probably doesn't fit all there. If you've got a problem with glycemia, it may be advantageous to exercise at a certain time of day. If you're a person with cardiovascular disease, we know that more people die in the morning of cardiovascular disease, usually upon waking when blood pressure changes, and maybe morning exercise is not indicated for you. So I don't think there's one size that fits all. And the questions that we ask in this review are basically, if you eat appropriately and time restrict eat, which I'll talk about in a minute, do you have to exercise, and vice versa? Is there an additive and synergistic effect? And we really don't know the answer to that question at the moment. So I'm not really going to talk any more about exercise, other than to say that a lot of the benefits, health benefits, that we will talk about in the next 30 minutes accrue from exercise, but ironically also accrue from this time-restricted eating, which, as I said, I'll talk about now. I think we missed a slide, but that doesn't matter in the interest of time. Circadian biology, we really have messed things up now. We have 24-hour access to food, to entertainment, to light, to just about anything that we want. And this wasn't always the case. And the guy on the right is Sachin Panda. He's, if you like, the father of popular science in this circadian biology area. And he's written a very best-selling book on the circadian code. I might add, it is a lay book rather than a scientific book, but again, as in all books like this, it over extrapolates some of the information. For example, the chapter on exercise is based on almost no references or no studies whatsoever. So we're still guessing a little bit here. The point that he makes, though, is, as this New York Times article says via the headline, when we eat or don't eat may be critical for health. And it may be more important than how much we eat. And that's a very provocative statement. So I'm saying here that the timing of nutrition may be a more important variable than actually what you eat. We've traditionally been brought up with nutrition and all the epidemiological studies saying, you know, high fat diets are bad, ketogenic diets are in, low carb. I mean, I'm not even going to get into that mess because it is a bun fight on Twitter most of the day with certain people advocating certain diets. I'm going to tell you that independent of diet, the timing of food and nutrient intake is very, very important. And this is really what happens when you disrupt some of the circadian rhythms by lack of daylight, disturbing sleep patterns, food intake changing, and exercise. You can see on the left-hand side there in blue is sort of a circadian alignment, a trace of one of the genes, the clock genes they're called, could be any number of them. And you can see there's a nice oscillation there in the pattern. You can see that in the right there, or in the middle of your screen, in the red or pink, there's a circadian malalignment. And the most common one that we've studied so far is shift workers. Shift workers disrupt their sleep, they disrupt their eating patterns, they disrupt when and if they exercise in the day. And what we get is what we call a time phase shift. So the peripheral clocks are out of sync, and eventually, if this persists long enough, then the central SCN is also out of sync. And there's now a lot of data, some of it convincing, others not quite so convincing, to show that this circadian misalignment, if you like, so disruption of normal circadian biology, has massive implications for health. And it's been linked to obesity, diabetes, heart disease, cancers, and a whole plethora of metabolic diseases. Now, the irony there is that most of those metabolic diseases are also linked to physical inactivity. And we shouldn't forget that during this talk. Although I'm not talking per se about nutrition, nutrition can just about prevent or at least help in the treatment of all these conditions. Tonight we're talking about nutrition, but I don't want to give the impression that nutrition is more important than exercise. So please don't take that away from this talk. Complicated slide, and on the right there's a lot of clock genes. It doesn't matter what they are, they're just alphabet soup, as my wife calls them. Just know that when you have a nutrient stress that disrupts circadian metabolism, and in this case it's a high fat diet, you can see that things start to unravel. So if you look at the rats on the left hand side of your screen there in the blue, you can see that there's all this inter-organ crosstalk when animals are free to exercise and they're on a normal chow diet. If you then disrupt their nutrition and place them on an extremely high fat diet, I might say not something that a human would ever wish to eat, but an extremely high fat diet, you can see via the wheel there that there's a loss of temporal relationships between some of the organs. The organ crosstalk is broken down. And that's partly because on the right, you can see that there's been these slight but subtle shifts in some of the clock genes. And that's a very important finding, that just bad nutrition, if you like, or nutrient stress in this case, as it's called, via a high fat diet, can actually disrupt the interorgan crosstalk and can also disrupt some of these very important clock genes. So that was, if you like, one of the first clues that nutrients are very, very important for calibrating or discalibrating, in this case, the circadian clock. And I'm going to really summarize a whole lot of animal studies here because I'm going to talk briefly about them, but I'm more interested in humans. I'm not interested in curing obesity in rats. I'm more interested in human diseases of disordered metabolism. But I'm summarizing a lot of studies here. And I want to introduce this term now, time-restricted feeding. That's what we use in animals, or time-restricted eating in humans. Now go back, and unfortunately I can't do a show of hands. If I asked you, as I did earlier on, for the time between your first and last meal, I would hazard a guess and say most of you are between 8 and 10 hours. And I've got no idea of seeing how many hands show that. But if you're in between 8 and 10 hours between your first meal and last eating occasion, that's pretty good, especially if you're exercising in the mix as well. If you're over 12 hours, you're starting to actually get into a danger zone. And if you're over 14 or 15 hours, in other words, the time duration between your breakfast, if you want to call it that, and your, I don't know, midnight snack, I guess, if it's that late, if that's 12 to 14 hours, then you probably want to think about altering your timing of your food. So in all the animal studies that have been done so far, when they restrict the daily feeding schedule to 8 to 10 hours a day and reduce it from a 14-hour day, you can see that there are all these positive benefits. The mice get leaner, their glucose homeostasis and insulin sensitivity improves, liver function improves, and inflammatory responses go down. Now, the interesting thing here is that these animals are still eating the same amount of calories. They're still eating the same energy intake. So we haven't done anything to the composition of food. We haven't told them to stop eating, you know, ice cream or whatever rats eat, certainly not ice cream, but we're not restricting foods. And this is, as you'll see later, the value of this diet. Most diets are doomed to fail, especially when you tell people to cut out certain foods. We're not doing that here. We're saying to the mice or the rats, just eat the same amount of food and just eat it over a shorter window. Now, complicated slide, but I really do want to walk you through this because this is, if you like, the proof of concept and the last animal study I'm going to show you of what all the human studies are based on. Now let's just go from the left-hand column down the page and then we'll switch to the right-hand column. You start with an obese mouse in the top left-hand corner. You feed them a high-fat diet, ad lib, nothing going on there. What happens? The mouse just gets morbidly obese. In other words, they get less healthy. You feed them the same crappy diet, a high-fat diet, but you restrict the window of eating to 8 hours. The phenotype of the mouse then presents as an obese mouse. Now, I'm not going to use the word fit because they weren't exercised, and fitness to me correlates with cardiovascular fitness and other parameters. So let's just say they were non-morbidly obese mice, but all they've done there is change the time of eating. In the middle panel there, they start with lean mice and they put them on both a high fat diet and a high sucrose diet. So a high sugar diet, you know, pretty typical of a Western diet. Again, they let the mice eat ad lib and they end up with an obese mouse. And then they restrict the same diet, the same energy intake, and you end up with a lean mouse. Now, those two studies, and I've summarized the studies below so you can see the references, they were done for 7 days a week. And when I got onto our human data, I'll tell you that people don't like to time restrict feed for 7 days a week. So what they did now is, in order to give the rats, you know, a couple of days off at the weekend, they asked the question, what happens if we just do Monday to Friday with the time restricted eating and let the animals just go bonkers at the weekend, just go out and socialize and do whatever animals do at the weekend, but eat ad libitum. And amazingly, they still got the same phenotype. They've still got a lean, fit mouse. Now, those findings are pretty incredible, I think. But the ones on the right are even more incredible. A wild type mouse is a mouse who's had no genetic manipulation whatsoever. So they're the normal, standard, you know, garden breed wild type mouse. What they did here is feed them both the same diets, and they fed them an ad lib diet, high fat, or they time restricted them. And again, the end output there is that you end up with a mouse who's had ad lib feeding, who's got metabolic disease, obesity, dyslipidemia. You can read all those things. On the other hand, the same diet, the same dietary composition, the same energy intake, the results with time restricted feeding in a healthy phenotype. Okay, nothing perhaps startling there, but the bottom slide on the bottom right-hand corner is, this is a clockless mouse. You've knocked out BMAL. You've knocked out one of the essential timekeepers in the whole body. So you're without the gene now that regulates some of the circadian biology. And what they did is the same study. They fed them the wild type or the clockless mouse, and they found that miraculously, even when you knock out a very important clock gene, BMAL1 as it's called, you still have a healthy phenotype independent of the gene that you've knocked out and independent of the energy intake of the diet. Now, of course, it's impossible to knock out those sort of genes in human studies. And of course, one gene doesn't control everything. If we were left up to just knocking a certain gene out and everything fell apart in the human, we'd be in big trouble. So obviously some of the other genes are compensating for that knockout. But even still, I think it's a pretty indicative proof of concept study to show that these mice lacking a circadian gene, clock gene, still have a healthy phenotype, even when fed a high fat diet when they time restrict their food. So enough about mice. I'm not really a mouse researcher, and we've got an article that's in review at the moment which actually looks at the translational value of animal versus mouse studies in this particular field. I won't bore you with that, it's still in review, but I will say just in passing, there are lots of differences. Mice are very small, 20 or 30 grams. Humans, especially a lot of Americans, are very big, 80 to 100, 150 kilograms. Animals voluntarily exercise. We voluntarily sit down. If you look at exercise metabolism, the differences between rodents and man is profound. We rely largely during exercise on exogenous stored substrates in the muscle, whereas animals rely a lot more on extracellular circulating blood glucose and blood lipids. Timing of food is obviously very different. And the fact that animals are nocturnal and exercise and eat at night versus man, who supposedly isn't meant to be doing that, are, I think, enough to give you perhaps an indication that everything that we find in the mouse isn't necessarily true for man or woman. So let's have a look now at what I think is a Watermark study. It's in a very, very good journal, but if you go back and read this, you'll find that there are a lot of problems with this. I think this was rushed to press very quickly. And as I say, it's a classic study. It's from Sachin Panda's group, and it was very innovative in that it used a smartphone app to track the eating patterns of individuals. And what they did, they had 156 overweight and obese males and females, and they monitored them for a period of 3 weeks. And they basically asked them to press this app every time they ate a food, so a certain eating occasion. They did a variety of other dietary methodology, which we won't get into now, but they were able to look at the eating occasions, the energy intake of these people, and when they ate. I'll get to the subject design on the right in a minute, but what they did after this is that they isolated 8 of these individuals, and I'll tell you who those 8 individuals were in a minute, and they gave them a 16-week intervention, and that was a time-restricted feeding intervention. I'm going to show you the next slide because this is called a feedogram, which I think is a lovely word. I've never actually heard of it. And although you can't tell there, and I encourage you to go and look at the paper, some 60% of energy intake was after 12:30 in the afternoon. And the average duration of eating, wait for this, was 14.2 hours, which may or may not explain why some of these individuals were overweight, obese. The eating window was 14 hours. Now, from this 156 cohort, they took 8 subjects, but it wasn't until I visited Sachin Panda's lab earlier this year, in probably my one and only overseas trip of the year, that I realized that they were all his lab members, which is a little sneaky. You're not really meant to have 8 of your lab members in a study, but nevertheless, they were. And this is them. You can see, let's have a look at the graph on the left-hand side. In the red is the time duration that a person ate. So let's look at, and I can't point to the screen, I feel fairly futile here, but you can see that that person started eating in the red, the extreme left red-hand bar, at about 7 o'clock in the morning and finished at about 9:30 at night. So a fairly prolonged eating window. You can see also from the red bars that people either start later and finish early, so there are individual differences. But I think what is striking is that you can see from the blue bars that in the case of these 8 subjects, who obviously knew what was going on because they were in Sachin Panda's lab, you can see that they restricted their time of eating quite significantly. So there were differences in the patterns of how they did it, but generally people started eating later and finished their last evening meal earlier. Now, after they'd done this for the 16-week period, they looked at body mass. And those of you in the field will know that the scales don't measure anything. They measure weight times acceleration due to gravity. Mass is a useless measure. They didn't have body composition in here. But what they did note is that after the intervention period, the 16 weeks of time-restricted eating, and then they followed them up again after 1 year, they'd lost 3.5 or 3.6 kilograms from memory, I think it was, and the weight stayed off. Now that's fairly incredible because most long-term studies don't show that. Now, it's just gone dark here because I haven't moved. Let me just move quickly. Here's the interesting thing. If you go back to the paper, you say, this is incredible. It is and it isn't, because if you go back to the paper, the average reduction in energy intake was around 20%. It varied between 14 and 22%. So is this due to time-restricted eating, or is it just the simple fact that they're just eating less? So this study can't tell you that. And although this has got hundreds of citations, I really encourage you to go and read the methodology. One thing I do like is the supplementary data in this particular paper in Cell Metabolism, and it will show you the fact that most of the what they call discretionary eating, that is the evening eating after dinner when people have, you know, chocolate or a wine or some ice cream or whatever it happens to be that is your sort of flavor of the month, it largely eliminated this discretionary eating. And that's probably where the loss of calories came from. So again, not really proof of concept. It's shown that time-restricted feeding is exactly that. They've restricted the time of feeding, but we don't know if that weight loss per se is due to the time-restricted feeding or actually due to the energy intake reduction, which is kind of glossed over in the paper. And any dietitian would have picked that up and probably given them a hard time, but editors of Cell Metabolism didn't see that. Now, again, I realize I'm giving you a lot of information here, and I've tried to put it as succinctly as possible. So I'm going to summarize this incredible study in one slide. This is a study where they followed up from the previous one and said, look, that was all very well and good, but was it the time-restricted feeding or was it the energy intake? So in this study, they took people and fed them in a clinical ward for 4 weeks. So they knew everything that went into their bodies. And there was no energy restriction here. They did baseline measures, they did calorimetry, they matched energy intake to energy output. So this is a true, very good dietary controlled study. That's the positive. The negative is this. This is what we call early time-restricted feeding. And the subjects in this study had their first energy intake at 8 o'clock in the morning, but had finished by 2 o'clock in the afternoon. Now, I'm laughing because, you know, if you're a student, you're not going to get a date on that sort of regime, I can assure you, which means basically at 2 o'clock in the afternoon, you've finished all eating occasions for the day. Now, again, as a proof of concept study, it's invaluable. And the point here is that everything was better afterwards. So time-restricted feeding has absolutely potent metabolic effects, and they are not due solely to the lack of energy intake. In other words, eating less calories. But again, whether you want to finish eating at 2 o'clock in the afternoon and be a social pariah, I'm not really quite sure. I certainly would not advocate that. So this is where we came in, and I'm going to show you a little bit of unpublished data here. We've got 3 or 4 studies actually in review at the moment on this. This is Sachin Panda here with one of my postdocs, Evelyn Parr. This is taken last year because she's since had twins, and I don't think she's even showing there. So this was a study that we set up to look at the feasibility of time-restricted feeding. We said, well, look, this is all well and good. These laboratory studies are great. The animal studies are fantastic. Proof of concept in a metabolic ward, blah, blah, blah. But what do people do in the real world? What about people with type 2 diabetes who are overweight, who are obese? Does time-restricted feeding work for these individuals? So this was the study, and again, this hasn't been published yet. We were looking at really the feasibility of time-restricted feeding in a cohort with metabolic disease. We gave them phone apps, we gave them cameras. They took incredibly 92% of every meal was photographed and sent to the researchers. You'll see why we haven't published this, because I forget how many tens of thousands of data points we have, but it was an incredible study, firstly in looking at the dietary side, but also showed you that these subjects were incredibly compliant. So we looked at their habitual diet. We said, we don't want you to change your diet. If you're eating crappily, well, so be it for the time being. That's not our problem right now. But then we wanted to look at a 9-hour window. Why did we choose 9 hours as opposed to 8 or 10? Because most people thought that this was probably a feasible thing that they could do. We had pre-study interviews with them, and they said, look, no, we can't do 7 or 8. We certainly can't finish eating at 2 o'clock in the afternoon. We've got families, we've got a social life, we've got kids, we have to do certain things. Now, how well did they do? Well, pretty good. On the top there is their control diet. That's their habitual diet for 3 weeks. And you can see it spans anything from, you know, someone's up at midnight there eating and still eating at the other end there in the dark blue or turquoise blobs there. You can see via the shaded area, that's the area that we really wanted them to eat in. And you can see they do reasonably well. Certainly the tails of those curves, or those plots, if you like, have come inwards. But you can also see there's some pretty naughty subjects who didn't do time-restricted eating at all. So this was, you know, a kind of interesting message for us. Time-restricted feeding in the real world is okay, but you've got to take into consideration that people do have lives, social lives, children, and the such like. And we did some validated questionnaires here, and we had a psychologist on board, and that's part of the paper that's in review at the moment. The bottom line was that most people perceived time-restricted feeding as being very, very beneficial. They liked things like the regimentation. They liked the fact that they were not told, you know, to cut out ice cream or to cut out Big Macs or this, that, and the other. What they didn't like is the fact that most of the time it meant a later breakfast and an early evening meal, and that just wasn't conducive to family life. So there are barriers to this. Let's just not put it out there as the, you know, the new diet miracle cure for whatever metabolic disease you may have. It's certainly feasible, we think, in at least in the short term, in patients with type 2 diabetes. We've also done, as I'll show you now, another study. This was a monumental study, and again, it looks very, very complicated. It was monumental because we took a group of overweight males. We had them do diet records. We then fed them a diet which we controlled. We fed them. So this is, if you like, a metabolic ward type of study, a proof of concept study. And this was a heroic intervention. We took biopsies throughout the day and night. We took 6 muscle biopsies at every 4 hours and sampled blood. The patients or the participants had continuous glucose monitors in, and we wanted to look at the effects of the time restriction versus what we called an extended feeding pattern there. So 15 hours versus a shorter 8-hour time restricted window on a variety of parameters. I'm just going to show you one tonight because I think this is a very interesting one. In essence, what we were doing, we were delaying the morning meal, in other words, the breakfast, and we were bringing the evening meal in earlier, as opposed to what we call the extended feeding, where they were just on their normal time course. But in this case, again, we fed them the food. So energy wasn't an issue in this study. We fed both of them. If you look at the summary of the graphs on the right here, the solid bars and the open bars there, you can see, I think, quite an important finding here from the venous blood glucose measurements. And remember, these are taken throughout the day and night. I won't bore you with the details, but we had a metabolic ward. We have a nursing department where we could have individual people that were all done at the same time. The doctor did, I don't know how many biopsies in the space of an evening, but lights were out. There were no cues. We did as much as we could to control the environment, as much as you can with someone coming and stick a needle in your leg at 2 o'clock in the morning. The patients were absolutely incredible. Some of them literally woke up for the biopsy and within 5-10 minutes were sleeping again. It was absolutely amazing to watch. The finding, which I find interesting, is the nocturnal glucose concentrations were lower with time-restricted feeding. As you could probably predict, if you finish your evening meal later, the time of fasting is longer. Now, why are nocturnal excursions in glucose dangerous? And they are, particularly in overweight, obese, and insulin resistant people and people with type 2 diabetes. Because during the night, if now we have the access to continuous glucose monitors, we see that a lot of these patients have very rapid excursions in blood glucose. You know, they get up to 9, 10, 11, 12 millimole in the night when they're just sleeping. Now, if we can dampen those excursions, which we know are linked to endothelial dysfunction and are precursors to cardiovascular disease, I think that's a very, very important finding. And again, it's not rocket science. You would predict that if you finished your evening meal earlier, then nocturnal glucose concentrations, in this case of glucose, should be lower. So I think that's an important finding. What I haven't shown you there, because it's under review at the moment, and it's been in the review for 3 months actually, but I won't mention the journal, is that we, with the biopsies, looked at muscle transcriptomics, so the genes in the muscle metabolomics, and also serum metabolomics. So we have a huge study on that at the moment with some very, very interesting findings on what time restricted feeding does to the, if you like, the metabolic machinery, and trying to find out and underpin some of the mechanisms which might confer the beneficial effects of time-restricted feeding. Now, I'm just looking at my watch thinking I'm going to wind up in a couple of minutes, Richard, so don't panic here. We started a little bit late.
Yeah, no
And
panic.
this is a question, I'm not easily panicked. This is a question that often comes up. Intermittent fasting. So there are many diets out there at the moment, you know, the 8-hour diet. You've got Michael Mosley in the UK advocating whatever he's advocating these days, based on very little data, I normally might add. Fasting, intermittent fasting. Are they all the same thing? I mean, isn't energy restriction and time restricted eating the same? It's not. It's definitely not. Intermittent fasting and prolonged fasting and energy intake are different to time restricted feeding. Time restricted feeding is really all about trying to get your circadian, your natural circadian biological rhythms back in sync. Intermittent fasting doesn't do that. In fact, it somewhat disrupts the circadian biology. So please don't think that fasting and time-restricted feeding are the same. And there's a couple of good articles, which I'm happy to send you, Richard, and if people want to, email in you afterwards. There's a very good couple of reviews on this in PNAS and a couple of other journals comparing fasting, intermittent fasting to time-restricted feeding. They definitely use different mechanisms for their, if you like, conferred improvement on metabolism. So why is time-restricted feeding good then? And again, this is from one of our collaborators, Paolo Sassoni Corsa and Gad Asher at University of California, Irvine. And what are the mechanisms, or at least some of the mechanisms, which may explain some of the positive effects of time-restricted feeding? Well, it's exactly what I've just said. It's really trying to align daily food intake with circadian rhythms. And in particular, now we're realizing that the gut microbiome plays a huge role in this. So there are daily oscillations in the gut microbiota, the production of long-chain fatty acids, and various other things that occur in the gut, which are very time dependent and time sensitive. So by coordinating your food intake to a pattern, and I'm not suggesting it always has to be 3 meals per day, but at least a regular pattern that you adhere to, is good for your gut. It may be one way where the gut, as we now know, talks to other organs and tissues via crosstalk, coordinates all this circadian rhythmicity. And the best example of how you disrupt this is just think of, 3 months ago when we used to travel, just go fly to America, fly backwards, fly forwards, go to a different time zone. What's the first thing you feel? Well, stomach's not quite right. You're eating out of sync. You're going to the bathroom out of sync. Yes, of course you are. You've disrupted the gut microbiome on a massive level. Not coincidentally, just anecdotally, even when flying 5-10 years ago, never eat all the meals on the plane that are provided to you. I used to get some very strange looks. Fly to LA for I'm eating. He used to come around and say, but Dr. Hawley, you've paid for this. It's like, I don't need it. Get to LA, 6 o'clock in the morning, have breakfast, right as a fiddle, particularly if you can get some sunlight and exercise as well. So there are lots of potential mechanisms whereby time-restricted feeding or time-restricted eating, if you're a human, may confer its benefits, but one of the ones that are worth exploring is the gut microbiome. And in the study that I just showed you with the diabetics, we have got data on gut microbiome. Unfortunately, it was just about to be analysed before the shutdown. So this is a paper which will be a long time in the making, I think. I can finish on this just very, very quickly, but this was an interesting study, again, which appeared in Cell Metabolism, one of the better ones to appear in there earlier in this year. And it looked at the gut microbiome, and it looked at an exercise training program. And just to cut to the chase and in the interest of time, it showed that those people who were responsive to exercise had a healthier gut microbiota. They had more diversity, which we generally associate with exercise and healthy individuals. They produced more short-chain fatty acids and the such like. And these people were able to be categorized as exercise responders or non-responders based on gut microbiota data, which is pretty incredible. I won't go into the animal study, but they took feces from the responders, put them in rats, made them exercise. And the humans with a healthy gut, when that was introduced into the animals, again, it took up the phenotype of the healthy human, and the exercise capacity of the rats was either up or down, depending on whether your gut microbiota was in a good health or bad health. So very interesting. So let's summarize. And I said, I've left some time for questions here, and I've got a lot of backup slides if there are questions that I can bring up. If you look on the left, I went through those very positive benefits of time-restricted eating in the animal, but we now are looking at a lot more human data coming out. And I may have missed some, but this slide is fairly up to date. I haven't put any mechanisms in there and any of our data, but there are a whole plethora of benefits now for overweight individuals, individuals with insulin resistance, pre-diabetics, type 2 diabetics and the such like, where some of these clinical markers are very much improved. And again, just to leave you on, and it's not published yet, but after just 2 or 3 weeks of the time-restricted feeding, we're getting drops in HbA1c, so glycosylated hemoglobin, which is a good marker of glucose control over the preceding month or 2 or 3 months, which are just astounding and are better than any medication. And again, the best feedback you can get at the end of the study is when the type 2 diabetic or the overweight person comes back and say, you've changed my life. And in some cases, many of the subjects came back in Evelyn's study and said, look, you've just changed my life profoundly. I've not only lost weight, but everything else is better. I feel better. I feel like exercising. All the clinical markers are great, but at the end of the day, the average person doesn't go around wondering what their HbA1c is. It's their daily living experiences. And I think for us, that was probably the most beneficial part of the study, just seeing how these diabetic subjects responded to this, how feasible it was. And yes, we got some nice clinical outcomes, but more importantly, the subject feedback and the feasibility of it was very, very high. So there's a list of our sponsors. You send me lots of money, you can have your name there too. I'll finish there. Richard, if there's any questions, I'm happy to take them. I'm not sure how you do this via Zoom. I guess you send them down the email or something like that, yeah?
Thank you, John. Really good talk, as always. How we do questions? Well, firstly, I wanted to get a little audio clip that did a round of applause at the end of the talk, but I ran out of time. As I was telling you, my 3-year-old gives me very little time to do anything, but that was great. And yeah, listen, there are lots of questions. I think we'll rattle through them in the order they came in, just to be fair. I've got maybe 2 pages worth of questions, but I'll be fair to the audience and let them speak. The first one is your good friend, from your good friend Eric Richter at Copenhagen. And he asked a question about the mice and time-restricted feeding in the mice. If they eat the same number of calories and end up with less weight or losing weight, then the time-restricted feeding, they must have increased the energy expenditure.
Yeah, I mean, yeah.
And otherwise, it doesn't make sense.
Yeah, absolutely. Greetings, Eric. I would expect a curveball like that from you, of course. Absolutely. And that is part of the thing in these studies. Some of them don't have metabolic caging. So, at least in the earlier work, some of the earlier work from Sachin Panda's lab and I think from the Dye lab as well, they've got an increased activity. So they're eating during a restricted window, which gives them more time to exercise. So their energy expenditure, in other words, their running distance in this case on a treadmill or sorry, not on a treadmill, on a running wheel is greater. So yeah, absolutely, their energy output has increased. That's not necessarily true in our humans, though. I might add, just as a backup to that, we put accelerometers on our human subjects, on our diabetic subjects, and also our overweight people in the 2 studies that I've described. We don't see an increase in number of steps in the day. These people aren't exercising, riding, running or anything, but we don't see a voluntary increase in the number of steps, which is interesting because the animal studies, as Eric's pointed out, tend to show that.
It's interesting, John. Sorry, I'm going to interrupt with a quick question on the back of that. So basically, and sorry if I missed some detail because I was noting some questions down for the audience, but it's important from what you're saying that it's the timing of feeding rather than the makeup of the feeding. So I was just wondering if you've got some data or you've come across the data that's manipulated the type of feeding, the macronutrients, for example, high fat, high carbohydrate, manipulated that with the timing restrictions as well, and you've seen any difference?
Okay, there's one study that's come out. I've actually got it as a backup slide. I mean, that wasn't prompted, but let me just, if I can,
it really wasn't.
I can feed through here. There's Michael Phelps, who's time restricted. Right, here's the study that's just come out in Nutrients now, actually. It's, again, I'll just cut to the chase. This is where they they didn't change the diet. That what they did is actually added extra protein to the diet. So this was the only study that I've got which looked at exercise and time-restricted feeding, and there are not many that do that at the moment. What they did here was resistance train these individuals, and they wanted to look if time-restricted feeding with caloric restriction. So it's not in the direct answer to your question because that's not been done to the best of my knowledge. But this one was increasing protein intake, reducing energy intake, ad-lib feeding of 14, 15 hours a day versus time-restricted feeding. And what they showed, I think in the next slide, and again, I'm happy to share slides with you later, is that they had a training adaptation, but nothing was different. So it didn't matter whether you time-restricted feeding with caloric restriction or you didn't time-restricted feed. In this case, you know, everything happened the same. So I'm not preempting a question, but I will preempt the question about the athletes. And again, I got asked this on Twitter a couple of times, you know, would you advocate time-restricted feeding for athletes? Well, not if athletes are training 3 or 4 hours a day and need to replace carbohydrate and muscle glycogen. No, they need all the eating occasions that they can get. On the other hand, if there are athletes who are making weight sports and the such like, then there may be a place for this. But a study that we're doing, actually the study that we're 98% finished, but we haven't finished the last 3 subjects at the moment, and I'll just very quickly talk about this, is one of the things that we're worried about with the energy restriction, both with the time-restricted feeding and certainly with the intermittent fasting, this day on, day off, is what happens to muscle protein synthesis. So we've got a study at the moment where we've used a technique using D2O labeled water and taken muscle biopsies from people who are either eating the same amount of calories, so the control, eating 33% less calories, time-restricted feeding, eating 33% less calories just across the whole 10-day study intervention, or after the 10 days, eating 33% less calories by fasting 1 day on and off. And our hypothesis there is that the prolonged fasting will reduce basal rates of protein synthesis. And we've shown this already in athletes that if you overnight fast them, and particularly if you reduce their energy intake only by as little as 500 kilocalories, you reduce basal rates of protein synthesis. And that for an athlete or anyone is terrible, because the prognosis for metabolic disease, and it's also linked to athletic performance, is lean muscle mass. So anytime you lose lean muscle mass, that's a very, very bad thing. So we'll have the answer to that, you know, next time we Zoom. Hopefully we don't. Next time it's a conference in a year, hopefully.
We can but hope. Moving on, got a question from Harry. Have any studies assessed circadian rhythms, i.e. gene clocks, pre and post time-restricted feeding interventions? I think
yes, ours,
already done that.
Ours is in review, so I can't tell you that at the moment. Yes, we've done that. You see shifts in a lot of the metabolites. The interesting ones are some of the branched chain amino acids and stuff are dramatically changed. And of course, some of the branched chain amino acids, high concentrations in the systemic circulation, have been linked to insulin resistance. So yes, you do. And they are improved with time-restricted feeding. So if you wait, I sent a letter to the editor the other day asking what had happened to our manuscript. If you wait a couple of weeks, I might have the answer to that, or at least I can share it with you, and it'll be out in the press.
Great. Thanks, John. Next question is from Chris Tyler, our very own Roehampton University. Does the makeup of the diet change when people are asked to change their behavior, i.e. when they are asked to time restrict and know what they are being, or know that they're being watched? Do their food habits change?
Yeah, that's a really good question. In several of our studies, we've actually fed them, so that takes that out of the equation. I guess the answer to that is people don't necessarily change the composition of what they eat, but they certainly, even when we follow them up 6 months or 9 months later, because some of these studies were done a year or so ago, they certainly, if I could pick one thing that they do much better, is this lack of discretionary eating at night, this comfort food eating after 7 or 8 o'clock at night. So most of them will say the biggest benefit that they've kept to and adhered to, which is probably why they've kept the weight off in most cases, is that they've knocked out, you know, the ice cream, the chocolate, the third wine, or whatever it happens to be. Whatever your poison is after 7 o'clock at night, that's the one thing that they've done. They don't seem to change the composition that drastically. You know, most of the type 2s for their glycemic control are on reasonably good diets anyway. I wouldn't say brilliant diets, because, you know, some of them were quite surprising when they came in, but Now, generally, in our studies, they can't alter their behavior patterns because we've clamped them, if you like. But when we let them go into the free world, which is really the litmus test as if your study's been any good or not, that's the one thing that I'll say. They stop their discretionary eating of foods, you know, nibbling during the evening or the late hours before they go to bed.
Great. Thanks, John. Next question is, do you have any insight regarding the tissue-specific benefits or adaptations of time-restricted feeding, specifically in insulin targeted organs, or sensitive? I think what they probably mean, insulin-sensitive organs.
Well, look, we've got, in that study that we did before, we've got OGTs and the such like, and we've got muscle data. So some of that we haven't even analysed yet, to be completely honest. I would suspect that time-restricted feeding will improve, you know, skeletal muscle insulin sensitivity. Absolutely, there's no reason for it to not. What it does to the liver, again, you know, if you've got a more prolonged fasting period, then the liver's just got to increase hepatic glucose output during a more prolonged period. So I think everything would be a positive, no matter what tissue you looked at. We've got, obviously you haven't got liver. We'll leave the Danes and the people in Stockholm and Denmark to do those studies, but we have got muscle data, and, you know, we've got a lot of this stuff which we still have to analyze, which is proving difficult at the moment while everyone's all over the place. So good question, yeah.
Great, John. I think I'll finish off with one from Lewis James, who's up at Loughborough University in the UK. Again, in the interest of time, he asks, in your opinion, is the time of the time-restricted feeding window important? Seems data with the most positive effects restricts eating in the evening.
Yeah, well, that's a really good question. There is one study actually which shows very little difference, a study from the Adelaide group, which Sachin Panda's on. I'm just trying to think of the first author. I think it's in Obesity Journal, where they looked at whether early time restricted feeding, and when I say early, it wasn't like the study which I showed you, the very extreme one. I think it was just shifting breakfast to 10 o'clock, starting later and evening meal later, versus breakfast at 12 o'clock and then eating later. There wasn't really that much difference. But again, I think it's very cohort dependent. It depends on what your, you know, metabolic disease or whatever you're trying to cure or improve happens to be. So at the moment, the practical recommendation, where, you know, we've given a few talks to the general public and the such like after our studies, has been that if you can delay your breakfast by an hour or so and bring your evening meal in, that seems to be the most practical. The only issue there is the evening meal timing. The earlier the better, but for practical reasons, school, sport, family, social occasions, that does prove, you know, tricky. One thing I forgot to mention, and I'll finish on this, is that in our study, we did the 5-2 with the diabetics. We let them eat what they wanted to on the weekends. And that model seems to be the preferred model. Although, you know, the animal studies have shown it worked, it was interesting to see that in humans, they gravitate almost to doing 5 days, but lose control, you know, over the weekend, which is fine. Although over the weekend, you would think that they would eat later at night and the such like, they didn't. So there were some really interesting findings. Again, how much you can extrapolate from a very small cohort of the clinical population to the general population, I'm not sure. We certainly need a lot more studies in this area with different populations, different feeding regimens, different starting times, etc. Yeah, good question.
Great, thanks, John. Sorry to those who asked questions I haven't had time to get to. I'll try and export the questions and put them somewhere with the video, which will be online hopefully in the next few days. I'm having some trouble with getting those up at the moment, so please bear with me. John, awesome talk. Really good to see you, and thanks for you know, taking the time out to, you know, to speak today. And you've obviously stimulated a lot of pun not intended food for thought. So hopefully, as you mentioned earlier, hopefully next time we see everyone, each other, it will be at a conference and face to face. But again, pleasure as always, and great hearing you talk. Just a quick reminder for those that are interested, we have Professor Eric Richter next Wednesday at the later time of 5 p.m. UK time. Just look out on Twitter for any updates on that. Again, thank you, John. Thank you for everyone for logging in, and I hope to speak to you all soon.
Thanks, Richard. You owe me a beer next time. All right. All happy?